Today's mini lecture is going to be all about the structure of DNA, and our key concept is that the DNA structure is the same in all organisms. So DNA is composed of four types of a molecule called a nucleotide. These nucleotides work together to make the two long chains that make DNA up. And each nucleotide has three parts as shown below. There is the phosphate group, the deoxyribose or the sugar, and then finally one of four nitrogen-containing bases. These nitrogen-containing bases are the only difference in all four of the nucleotide types. So basically, the nitrogen bases can be either pyrimidines, which means single ring, or purines, which means double ring. The pyrimidines include thymine, T, and cytosine, C, and then the purines are adenine, A, and guanine, G. So two scientists, Watson and Crick, are generally credited with the discovery of the structure of DNA. And what they did is they built a variety of 3D models using clues, just like you guys did, until they kind of figured it out. So they realized that DNA had to be something called a double helix or a twisted ladder. That's the overall shape. And the outsides are what we call a backbone made out of sugar and phosphate bonded together. And on the inside, you have bases that are bonding together. Now, Watson and Crick couldn't have done this alone. They built on work from other scientists. One of those two scientists was Rosalind Franklin, and she was the first person to take X-ray images of DNA, which are shown right here. So her X-ray images basically suggested that DNA was a double helix or a twisted ladder, and the width was the same all the way across, 4 molecules, and you can kind of see that in here. This is like a picture of DNA from above. Then Erwin Chargaff also did a bunch of experimenting and created Chargaff's rules, which basically state that if you were to take DNA apart, rip it in half, take off every adenine, thymine, cytosine, and guanine, and put them in piles, the amount of adenine is going to be equal to the amount of thymine, and the amount of cytosine is going to be equal to the amount of guanine. Now, nucleotides are going to always pair in the same way, and we call these base pairing rules. So when the bases pair together, adenine is always going to pair with thymine and thymine with adenine. Cytosine is always going to pair with guanine and guanine with cytosine. Now, because they pair in this way, and it's always going to be one of the pyrimidines with one of the purines, the double helix is always going to have the same width, 4 molecules wide. So what this picture is going to show, this is a ladder model, and we use this sometimes so we can look at the molecular structure without the twisting. And if you look at this model, you'll notice that the backbone or the side chains are made of a phosphate group and a deoxyribose sugar, and those are held together by these really strong bonds called covalent bonds. Covalent bonds involve actual electron sharing between the two, so they're a very strong bond, very difficult to break. The bases in the middle, however, are connected by these things called hydrogen bonds, and hydrogen bonds are very weak and can be easily broken. And this is going to come in really handy when we talk about replication and transcription later on. You'll also notice that A always pairs with T and T with A, C with G, G with C. You'll also notice that you've always got a pyrimidine with a purine. And then one final thing you'll notice is that when T pairs with A, there are only 2 hydrogen bonds, but when C and G pair together, there are 3, and you will always see it in this way.